Asymmetric carbon is having 4 valencies. Carbon is having how many valencies? 4 valencies. 4 valencies are there. 2, 2. 4 valencies are there. And we found that if carbon 4 valencies are occupied by different group H, R1, R2, R3, then this is asymmetric carbon. Yes, when there is asymmetric carbon atom, then the compound will show optical isomerism and stereoisomerism. Again I repeat, any compound having asymmetric carbon atom will show optical and stereoisomerism. Again I repeat, yes, glucose, look at the glucose. If I say carbon number 2, in this one group is OH, second group is H, third group is this large one, fourth group is this CHO. So carbon number 2, if I say it is having 4 valencies, all 4 valencies are occupied by OH, H, CHO and this large one, different group, asymmetric. C2 is asymmetric. C3 is also asymmetric. One group is H, second is OH, third is this having CHO, fourth is this. So C2 is asymmetric. Even if you don't remember why it is symmetric, okay, but have an idea. Yes, C1, C2 is asymmetric. I make it circle. C3 is asymmetric. C4 is asymmetric. C5 is also asymmetric. Okay, so these are asymmetric carbon atom and 4 asymmetric carbon atom in glucose, while in fructose there are only C3, C4, C5. C3 is asymmetric, C4 asymmetric, C5 asymmetric. So 3 asymmetric carbon atom. And we found that we can calculate the number of stereoisomers. What I said? Stereoisomerism. What is stereoisomerism? Means orientation in space. We found that suppose I am having these two things, one this pen and second is this, yes, this key, pen and key. Yes, we found that if carbon is having, I am carbon, black t-shirt, okay, I am having carbon, having HOH. If OH is at this side, the compound is having different property, but if OH is at this side, the compound is having different property. Do you agree? Yes. If OH is at this side, yes, I can't write from my left hand. I can write with my right hand. Okay, so we can remember like this. But means we found that if OH orientation changes around the asymmetric carbon atom, the property of compound changes. So stereoisomerism means orientation around asymmetric carbon atom. Stereoisomerism means orientation around asymmetric carbon atom, compounds which have same structure formula. Yes, same structure formula would be there, but differ only in spatial configuration, means configuration orientation in space, known as stereoisomerism. Stereoisomerism may be of 3 types, in fact multiple types, but 3 types are frequently asked. One is, yes, you will write here only, that is enantiomerism. That is capital D and capital L isomerism. Capital D and capital L isomerism. Enantiomerism is also called capital D and capital L isomerism. Okay, what is capital D and capital L isomerism? Suppose I give you this glucose molecule. In this glucose, carbon number 5 is asymmetric. One group is OH, second group is H, third group is CH2OH, and the fourth group is this large one. So we found that in this glucose molecule at carbon number 5, OH is on right side. OH is on right side, and we said that this is capital D glucose. Capital D glucose. C5, yes, also called penultimate carbon. Sometimes we say C5 is penultimate. Ultimate means last. Penultimate means previous to last. Last is carbon number 6. Previous to last is carbon number 5. If OH orientation at carbon number 5 is on right side, then it is capital D glucose. But if OH orientation at C5 is on left side, then it is capital L glucose. Capital L glucose. And we found that if I put a mirror, between these two, compound, this is capital D glucose, capital L glucose. If I put a mirror, yes, let me take this color. If I put a mirror between these two, you will see these are exactly mirror image. These are accurately mirror image. Yes, look at the screen and see. Yes, if OH is on right, yes, in this OH is on right side, in this OH is on left side. Mirror image, mirror image, mirror image, mirror image. So you will say, sir, OH orientation is different at each and every carbon. OH orientation is different at each and every carbon. Yes, OH orientation is different at each OH at each and every carbon. Okay. Yes, there was somebody disturbing outside, so that's why. Yes. But we defined capital D and capital L on the basis of C5. At C5, if OH is on right side, capital D glucose, on left side, capital L glucose. So I change the color. ग्लूकोन डी जान में जान डाल दे पीते ही. ग्लूकोन डी जान में जान डाल दे पीते ही means in our body all carbohydrates are found in capital D form. Yes, question is asked. This is a favorite question. This was favorite question of PGI Chandigarh. Yes, glucose. Yes, in our body. Yes, you can write in our body. But after that I will take break. In our body all carbohydrates are found in capital D form. Yes, how to remember? Mnemonic is glucon D. Jaan mein jaan dal de पीते ही. So why we are so much concerned? Why they are asking question again and again? If I give you a molecule of D glucose, it will be absorbed, metabolized, will give you 32 ATPs. If I give you capital L glucose, it is not metabolized. If I give you capital L glucose, it is not metabolized. Means the slight change in the orientation changes the complete property. And how you will remember in our body carbohydrates are found in capital D or capital L? By the mnemonic glucon D. Okay, glucon D is having vitamin D. It may be supplemented, but glucon D means that this glucose is present in capital D form, which can be metabolized, which can produce 32 ATP. Got the point? Yes. So in this enantiomers, हिंदी में मैं कहता हूँ आईना, enantio आईना, ऐसा कुछ pronunciation सा आता है. Mirror, wherever you find the mirror, without second thought you will say enantiomerism. In NBA question was asked, they gave two structures and they put mirror in between these two structures. Yes, and they ask, yes, what is this? This is enantiomerism. Yes, sometimes they open the book like this and they say these are mirror image. Yes, this is again enantiomerism. So enantiomerism or capital D and capital L isomerism, they are mirror image. So in picture MCQ, wherever you find mirror image or wherever you find the mirror in the image, without second thought, answer would be enantiomerism. So stereoisomerism, I discussed orientation, OH orientation at penultimate carbon C5 and all carbohydrate in our body found in capital D glucose. Sorry, capital D form. Stereoisomerism. Stereoisomerism may be of many types. Three types common on which questions are asked. One is enantiomerism, second is epimerism, third is anomerism. So first I discussed enantiomerism. Enantiomerism means OH orientation at penultimate carbon. Whenever there is OH orientation change at penultimate carbon, means if we define, although in enantiomerism they are mirror image, OH orientation is different both in not only on penultimate, other carbon also OH orientation is changed. But we defined capital D and capital L on the basis of penultimate carbon. If penultimate carbon OH is on right side, that is capital D, left side capital L, and they are enantiomers, that is capital D and capital L isomerism. Okay, we found that capital D and capital L isomerism, all carbohydrate in our body are found in capital D form. Now, if I ask you amino acid, this amino acid is having 4 groups. This amino acid is having 4 groups. All 4 groups are different. So amino acid will also show optical and stereoisomerism. As wherever asymmetric carbon atom is there, it will show optical and stereoisomerism. So amino acid will also show optical and stereoisomerism, and amino acid will also show enantiomerism. Amino acid will show enantiomerism. Yes, and we will write here, yes, all amino acid in our proteins are found in capital L form. Capital L form. Yes, the glucon D, the purpose was to make you remember glucon D, carbohydrate in D, amino acid in capital L. Clear? But this question was favorite question of PGI Chandigarh. Why I am mentioning PGI Chandigarh? Wherever I mention PGI Chandigarh, that is potential question for INICET. Recently, whatever question they ask in INICET, they were previous year question of PGI Chandigarh. Because in PGI Chandigarh, there was, it used to be a separate segment of molecular biology, and in biochemistry was little bit more weightage. So, whatever question of, yes, PGI Chandigarh is important for INICET. So in PGI Chandigarh there was a question, yes, all carbohydrate in our body are found in capital D form. This statement is true. There were multiple correct answer they used to be in that exam. This second statement they used to give, all amino acid in our body are found in capital L form. This statement is true or false? Hear carefully and tell me the answer. All amino acid in our body are found in capital L form. This is true or false? This is false. This I said all amino acid in our protein, not body. Yes, I will discuss that amino acid join to make peptide bond and protein. So all amino acid in our protein, when amino acid join to make protein, they join in capital L form. But if they are free, they are usually found in capital L form, but there are few amino acid in our body which are found in capital D form, just like amino acid D-serine, D-serine in brain, found in capital D form. You can, many times I say that yes, if you find the word, you will find the relation and answer. Just like D, serine, brain कहाँ पे होता है? सर में होता है, खोपड़ी में होता है. D for दिमाग, तो डी सेरीन कहाँ पे है? ब्रेन में. ओके, दिमाग, सर, ब्रेन. ओके, यस. NMDA, NMDA, N-methyl D-aspartate. N-methyl D-aspartate means that aspartate is present in capital D form. This is excitatory neurotransmitter. Excitatory neurotransmitter. Clear? Now, so this is enantiomerism. So I integrated this. Yes, question is asked, all amino acid in our protein are found in capital L form, means whenever amino acid will join, it will always join in capital L. But if it is free, it may be found in capital L or D. But most commonly they are found in capital L form, but there are few amino acids just like D-serine and MDA which are found in capital D form. Clear? Now, so this was enantiomerism. So first type of stereoisomerism is enantiomerism. Now, second type of stereoisomerism is epimerism. Epimerism. What is epimerism? Epimerism means OH orientation. I don't write here. I can write, if you have space here, so you can write here only. OH orientation at other than penultimate carbon. Penultimate carbon. OH orientation at other than penultimate carbon means other than C5. If OH orientation is changed, that is epimerism. Yes, if I write here glucose and here write mannose. Okay, glucose and mannose, look at the screen. If glucose at carbon number 2, OH is on right side. This is glucose. At carbon number 2, if OH is on left side, this is mannose. So glucose and mannose, the only difference is OH orientation at carbon number 2. Look at me. Suppose this is glucose having 6 carbon atom, 1, 2, 3, 4, 5, 6. Yes, glucose at carbon number 2, OH is on right side. I hold this OH and rotate this OH to left. It will become, comment, it would become mannose. Again I repeat, this is glucose. In this glucose, 1, 2, 3, 4, 5, 6, carbon number 2, OH is on right side. If I hold this OH and rotate this OH to left, it will become mannose. So means only difference between glucose and mannose is OH orientation at C2. At C2 OH right side, that is glucose. At left side, this is mannose. So we say mannose is epimer of C2 at, mannose is epimer of glucose at carbon number 2. Question is asked, mannose is epimer of glucose at C2. Question is asked, mannose is epimer of glucose at C2. Very good. You have given correct answer. Now, look at the screen and write, this is glucose. This is very, very important. You can't afford to forget this. This is glucose, this is galactose. This is galactose. This is glucose, this is galactose. If I say that at glucose at carbon number 1, 2, 3, 4. Carbon number 4 OH is on right side, this is glucose. At carbon number 4 OH is on left side, that is galactose. So glucose and galactose, everything is same, only difference is OH orientation at C4. At C4 OH on left side, this is galactose. Now again look at me, this is a pen. Suppose this is molecule of galactose. This is a molecule of galactose. 1, 2, 3, 4. Carbon number 4, OH is on left side. I hold this OH and rotate this OH from left to right. It would become glucose. Means that is galactose, and this galactose at carbon number 4 OH is on left side. What I do, I hold this OH and rotate this OH from left to right side. It would become glucose. So galactose and glucose, yes, they are apimers. They are apimers at carbon number 4, and we all know glucose is normally metabolized in our body. I told you glucose metabolize glycolysis like this. And galactose metabolism, yes, we also want to produce energy, because when child is born, the staple food is, or the only source of energy is lactose. I will discuss lactose is made up of glucose and galactose. Means a child is born, child is taking, it is taking whatever you say. Yes, child is taking lactose. Lactose include galactose. So galactose, we want to utilize galactose. So there should be a separate pathway for galactose. But nature thought, why we make separate pathway for galactose? What we will do, we will take galactose at carbon number 4, we will hold OH and we will rotate this OH to right. Galactose will be converted to glucose, and then glucose will be utilized. So galactose metabolism is nothing but conversion of galactose to glucose. Yes, point it out. Point it out. Don't turn your pages. Don't write anything. Just let me tell you. Yes, galactose metabolism I will discuss at that time. At that time I will tell you what is galactose metabolism. In galactose metabolism, yes, I will discuss this galactose metabolism. Galactose metabolism is nothing but galactose is converted to glucose. So now you will never forget, galactose metabolism is nothing but conversion of galactose to glucose, because we know glucose can be metabolized by glycolysis. We have all the enzymes for glycolysis. There should not be any other pathway for galactose metabolism. What we will do, we will take a galactose, we will rotate this OH from left to right. But suppose this is galactose molecule, I hold this OH and rotate, the complete molecule will rotate. So that's why first of all I will take galactose, I will put energy, galactose to galactose 1-phosphate. I will use some energy. With the energy I will fix this galactone is UDP. My hand is UDP. I attach this galactose to UDP. Now I hold this OH and rotate this OH to right. Now this galactose molecule will not move. So this gesture will help you in your exam. Yes, I said I took galactose metabolism nothing but conversion of galactose to glucose. How I converted, I give energy galactose 1-phosphate, I attach galactose to UDP galactose, then I rotated OH from left to right. UDP galactose will be converted to UDP glucose, and this glucose will be released. Did you understand this? Please do comment. So I will discuss galactose metabolism in detail. What are the defects in galactose metabolism? For the time being, you understood what is the galactose metabolism. Question was asked. Yes, question was asked and you will tell me the answer. Yes, that question was asked. When this question was asked, that question considered to be galactose metabolism question, but exactly the question was epimerism. Let me show you. Question was asked, which of the following Which of the following enzyme is not involved in galactose metabolism? What was the question? Which of the following enzyme not involved in galactose metabolism? Option A, galactokinase involved. Option B, galactose 1 phosphate uridyl transferase involved. Option C, UDP galactose 4 epimerase. And option D was UDP galactose 2 epimerase. The answer would be, yes, you all know galactose is epimer of glucose at C4. So in that question, even if you don't remember the exact name of enzymes, it's okay. But if you remember, this galactose is epimer of glucose at which carbon? C4. So enzyme involved would be UDP galactose 4 epimerase, and UDP galactose 2 epimerase was the answer. UDP galactose 2 epimerase is not the enzyme of this galactose metabolism. Very good. So this is the, so don't worry, I will discuss all these things. Usually I say the first day in biochemistry is base. If you will remember like this, you will never forget. Whenever they will ask question, you will be able to solve question very easily. Okay, we are integrating things. So glucose and galactose, yes, that we say distereomers. That is distereomers, mannose and galactose. Okay, I will take care of those doubts in later, okay, part. Yes. So glucose and galactose epimerism. Clear? So this is epimerism. Third type of isomerism is, stereoisomerism. I am talking about stereoisomerism is anomerism. What is anomerism? Yes, anomerism means, yes, you all have seen whenever in nucleotide we draw the structure of pentose sugar, ribose sugar, we always draw in a cyclical form. So we saw any carbohydrate having 5 or more than 5 carbon acquire a cyclical structure. So any carbohydrate having 5 or more than 5 carbon acquire a cyclical structure. So glucose will acquire a cyclical structure, fructose will acquire a cyclical structure, and we found that when glucose acquire a cyclical structure, this C5 bind with this aldehyde. C5 bind with this aldehyde. So functional group of glucose is aldehyde. We found that penultimate carbon bind with C1. But in fructose, penultimate carbon will bind with functional group. Functional group is present in fructose at, yes, fructose at C2. Okay, C2. So in yes, so we found that glucose and fructose both can acquire a cyclical structure. When glucose acquire a cyclical structure, C5 bind with C1 because the functional group of glucose is C1. Fructose C5 bind with C2, yes, because functional group of fructose is keto group. Yes, I will solve a very important question. Yes, C2. Very good. Now, if I say there is a ring structure formation, yes, you will find that in ring structure formation, yes, what would be the ring structure of glucose? Yes, this is the ring structure of glucose. In this, C1 bind with C5 with the help of oxygen, yes, aldehyde group, and we said that this ring is 1 2 3 4 5 carbon, 1 oxygen, that is pyranose ring. Which ring? Pyranose ring. Pyranose ring is a 6-membered ring. How many atoms are there? 6 atoms consisting of 5 carbon atom and 1 oxygen atom. Clear? Pyranose ring is formed by reaction of C5 carbon hydroxyl group with C1 aldehyde group. Yes, C5 to C1. Clear? So this is a pyranose ring. But if I modify it, you can also do that. Yes, you can also do that. Yes, this is glucose. I write here glucose. Yes, so glucose is usually found in which ring? Pyranose ring. But if I say C1, C2, C3, I am modifying deliberately. C1, C2, C3, C4, C5, C6, cut. So I cut this, and now yes, this is C6, C5, C4, C3, C2. Being cause in fructose, yes, C2 bind with C5, that's why this is called furanose ring. Furanose ring. Furanose ring is having 5 atoms and 4 carbon plus 1 oxygen. Clear? So fructose is usually found in F4 fructose, F4 furanose, while glucose is usually found in which ring? Pyranose form. So glucose is usually, not always. Okay? Glucose, most of the glucose molecules are found in which ring? Pyranose ring. Yes, fructose is usually found in furanose ring. Clear? Even if you don't remember so much of detail what I said, yes, if you remember it's good, but if you don't remember, just remember, yes, there are two types of ring: pyranose ring, furanose ring. Nobody is asking which is C1, which is C6. If you remember that functional group take part in the ring formation. What I said, functional group take part in ring formation. After ring formation, if I say this is glucose, in this glucose carbon number 1, after acquiring cycle structure, this is H, this is OH. First group is H, second group is OH, second group is OH, third group is this, fourth group is this. This carbon one is symmetric or asymmetric? Please do reply. Yes. Yes. Let me repeat it. Furanose ring. In furanose ring, there are 5 atoms. There are 5 atoms. This ring is having 5 atoms: C2, C3, C4, C5. 5 atom, and this is oxygen. So 4 carbon and 1 atom of oxygen. So out of 5 carbon. So just like we said, pyranose ring is a 6-membered ring, but furanose ring is a 5-membered ring, having how many carbon? 4 carbon. How many oxygen? 1 oxygen. So furanose ring is present in fructose, having 5 atoms. Understood? Okay. And question is asked, glucose is mainly pyranose, fructose is mainly furanose. And if I say this is pyranose ring of glucose, in this glucose, after acquiring cyclic structure, this C1 becomes asymmetric, and any compound having asymmetric carbon atom will show optical and stereoisomerism. Now look at me. Suppose this is a cyclic structure of glucose. This is a cyclic, my hand, this is cycle structure of glucose in a plane, and if I say in this plane at carbon number 1, if OH is below the plane, then it is alpha glucose, and if it's above the plane, this is beta glucose. Means OH is below the plane, yes, this is alpha glucose, and OH is above the plane, this is beta glucose. So alpha glucose and beta glucose. So that's why anomerism is also called alpha beta isomerism. Alpha beta isomerism. Yes, you will remember OH below, B matlab below, but B not beta. B is below the plane, but it's not beta, it is alpha. Above the plane, yes, beta glucose. So what is the importance of this? Yes, how questions are asked, let me discuss here only. Yes, we have space, little bit space here. Yes, if you have space or next page is totally vacant, yes, so many space, so much of space is there, and in fact I have given space here. Yes, now a question is asked, glycogen is a polysaccharide made up of glucose, 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 glucose. Glycogen is a polysaccharide made up of so many glucose molecules. Glycogen I have discussed, made up of so many glucose molecule, you all know, and these glucose are joined by alpha 1,4 linkase. Which linkase? Alpha 1,4 linkase. Starch, again glucose molecule are joined by alpha 1,4 linkase. Alpha 1,4 linkase. Cellulose, again glucose molecule, these are joined by alpha 1,4 linkase. No, this is beta 1,4 linkase. So glycogen is a polysaccharide of glucose, 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 glucose. Starch is a polysaccharide of glucose, 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 glucose. They are joined by which linkase? Alpha 1,4 linkase. But cellulose, these are joined by which linkase? Beta 1,4 linkase, and we all know that cellulose is not digested in our intestine. Cellulose is not digested in our intestine because there is no enzyme which can cut this beta linkase. Now look at me. Means this is glucose. If I take one molecule from glucose, yes, starch, if I take this molecule of glucose, yes, this molecule of glucose of starch, in this glucose you will find OH is below the plane. OH is below the plane. But if you talk about cellulose and you take glucose, in this glucose OH is above the plane. Means OH orientation changes. If OH orientation is below the plane, it is starch is utilized in our body. Cellulose, OH is above the plane, not metabolized. So that's why we say alpha beta anomerism is important. If OH is below the plane, metabolized. OH is above the plane, not metabolized. So question was asked in AIMS that cellulose is not metabolized. Cellulose is not metabolized in our body due to beta anomerism at C1, carbon number 1. Yes, cellulose is not metabolized, or digested, better to say digested, not digested. If I say metabolized, it's also correct, but better to say not digested, not digested in our intestine due to beta anomerism at which carbon? Carbon number 1. At carbon number 1, yes, if OH in glucose, OH is below the plane, alpha glucose metabolized, above the plane not metabolized. But they did not ask question in such a simpler way. They asked question little bit difficult. The question was, yes, Cellulose is not digested in our intestine. It acts as a dietary fiber. Cellulose is not digested because option A, anomeric C1. Option B, anomeric C2. So answer was yes. Actually they should mention that we are talking about alpha beta, but they did not mention alpha beta. You must know anomeric means alpha beta isomerism. You know that cellulose is polymer of glucose. Glucose will show anomeric at functional group, and functional group of glucose is aldehyde, which is present at carbon number 1. So I can say anomerism is shown at functional group. Yes, functional group. Yes, shown at which group? Functional group. Yes, clear? Got the point? So fructose will show anomerism at C1 or C2? C2. So next time jip mein ask question that inulin is a polysaccharide of fructose not digested due to anomeric. Option A, please give answer. Yes, fructose is polysaccharide made up of fructose not digested due to option A, anomeric C1. Option B, anomeric C2. Yes, anomeric C1 or anomeric C2. तो आंसर was yes, anomeric C2. Okay, so you are getting this. So let me repeat it fast. I know that it is taking time, but let me summarize this page before moving further in 5 minutes only. Carbo means carbohydrate means carbo means carbon, hydrates means water, carbohydrate CH2N. This is the empirical formula of carbohydrate, C6H12O6, that is glucose. Glucose is a prototype monosaccharide. Glucose, fructose, galactose, mannose all are having same molecular formula C6H12O6. You can see that glucose is having C6H12O6, fructose is having C6H12O6, mannose is having C6H12O6, galactose is also having C6H12O6. They all are having same molecular formula, so they are isomers. We said that glucose is having aldehyde group at C1, fructose is having keto group at C2. In glucose there are so many OH group, and whenever there is OH group provide polarity. OH group has tendency to attach with phosphate. We found that out of 20 amino acid, wherever amino group is there positive charge, acid group is there negative charge. But amino acid, thus yes, we define amino acid on the basis of side chain, and we found that there are 3 amino acid which are having OH group in their side chain. Those are tyrosine, threonine, serine. So we know that wherever OH group is there, they has tendency to attach with phosphate, and they are polar. So these 3 amino acids are polar. This question was there, yes, if yes, protein acquire more tendency to be phosphorylated, means the alanine has been replaced with serine. Now we found that any carbon having all valencies are occupied by different group, that is called asymmetric carbon atom. Asymmetric carbon atom will show optical and stereoisomerism. Amino acid will show optical and stereoisomerism. Yes, glucose will show optical and stereoisomerism. Fructose will show optical and stereoisomerism. What is stereoisomerism? Stereoisomerism means OH orientation or orientation of different group in the space. We found 3 types of stereoisomerism are important. Enantiomerism, ayna, mirror. Wherever you find mirror image, and you will remember enantiomerism is also called capital T and capital L isomerism, that is seen at penultimate carbon. At penultimate carbon, OH is on right side, capital D glucose, on left side capital L glucose, and mnemonic is glucon D, jaan mein jaan dal de peete hi, glucon D. All carbohydrate found in capital T. For amino acid also show enantiomerism. All amino acid in our protein found in capital L form, and few amino acids are there in our body, just like D-serine in brain, hence NMD NMDA neurotransmitter are found in capital D form, and enantiomers are mirror image. Question has been asked. Epimerism means OH orientation at other than penultimate carbon. Yes, if OH orientation C2 OH group on right side, yes, that is glucose. Left side mannose, glucose. C4 OH is on the right side, galactose OH is left side. If we want to metabolize galactose, no need to keep separate pathway, just convert galactose to glucose. We attach energy with galactose, we fix galactose in the UDP is my hand, and now you become UDP galactose. We hold the OH, we convert into right UDP glucose is metabolized. So galactose metabolism is nothing but conversion of galactose to glucose. So this is epimerism. Yes, we usually say that galactose and mannose, they are distereoisomerism. Okay, distereoisomers. Okay, epimerism, you will remember only one carbon. Yes, we will say that epimerism is only at one carbon. Okay. Anomerism, alpha beta isomerism. Yes, anomerism is shown at functional group. Anomerism is shown at functional group. Glucose will show anomerism at C1, fructose will show anomerism at C2. Okay, now glucose mostly found in pyranose ring. Pyranose ring is a 6-member ring. Question has been asked long time back, Jipmer question. Okay, not repeated multiple time, but yes, you must know that implementation they have asked about anomerism. What is anomerism? Means any carbohydrate having 5 or more than 5 carbon atom acquire a cycle structure, and after acquiring cycle structure, the functional group become asymmetric, and when functional become asymmetric, it will show stereoisomerism. If OH is below the plane, alpha; if OH is above the plane, beta. And we found that that beta linkage is not digested in our intestine. That's why cellulose is not digested in our intestine. That's why we say cellulose is not digested because of beta anomerism at C1. Yes, and yes, inulin is not digested in our body because of beta anomerism at C2, because fructose show anomerism at functional group carbon number 2. Okay. So these are 3 types of stereoisomerism on which frequently questions are asked, and they are having import.
Hi friends, so like, share and subscribe. If you have any doubt, comment below. Thank you.